Good morning. Thank you all for coming today. My name is Sadie Paez, and I am the Program Director for Genome 10K Vertebrate Genomes Project. This is, of course, a thrilling day for all of us from the Genome 10K Consortium and for our sequencing hubs at the Vertebrate Genome Laboratory at the Rockefeller University, the Wellcome Sanger Institute in the UK, and the Max Planck Institute of Molecular Cell Biology and Genetics and Center for Systems Biology in Dresden, Germany. For the benefit of the journalists in the room today and joining us via live stream, I want to give you a few details as to how we are going to proceed, and then I will turn the podium over to the Genome 10K chair, Dr. Erich Jarvis, who will present our announcement. We will then have brief statements from our Genome 10K co-founders, council members, and sequencing and assembly hub directors. We will have about 20 minutes for Q&A, which will include additional panelists seated in the front row that represent other Genome 10K council members, genome assemblers, and database directors. Our technology partners are also in the audience and available to address questions. For our live streaming audience, please use the link to submit questions. Following the press conference, our panelists and technology partners will all be available for one-on-one or group interviews, photos, and videos. You can join us at the tables in the back or in the adjoining rooms. The press packet includes a list of available interviewees. Thank you. I would now like to invite our Genome 10K chair, Dr. Erich Jarvis, to the podium for our announcement.
Thank you, Sadie. Yes. All right. Well, folks, this has been a long time coming. We have familiar faces up here, also in the audience, as well as the public media and editors from journals and technology partners. What I'm going to announce today is a project that the Genome 10K Consortium has been working on for a while now, actually for 3 years. Quietly, sometimes you see some announcements here and there, but never have we come together at a moment like this to say something more publicly, to take a stake in the ground for something that we've been working on. And that's the purpose of this gathering here now. So what I'm announcing here is the Vertebrate Genomes Project, organized by the Genome 10K Consortium, and some species that we're actually going to release in the next 24 hours in terms of their genomes. I have to give a little bit of history here of how we got where we are at before announcing the species. So this organization began back in 2009 with the three leaders up here, David Haussler, Steve O'Brien, Ali Ryder, in this picture up here, as you can see, where next-generation sequencing technology was just taking off the ground at that time after the Sanger approach was used for the human genome. And they organized a conference back in 2009 with the goal eventually of sequencing 10,000 species and published a paper on this goal. And a number of projects have occurred since that time that the G10K organized or helped facilitate. One was an assemblathon competition. Find the best methods to assemble a genome properly, because we already knew at that time there were lots of errors in them. Another assemblathon competition using real genome data. The first is on simulated data, on the parrot species and a few others. Also an online-a-thon competition. Once you have the genomes, you got to align them together to figure out who is similar to who and who is related to who. So they formed that. And eventually, 101 Genomes Project in collaboration with the Beijing Genome Institute. And out of that last one, 11 of those genomes were used for, if the folks behind here, if they want to look, it's okay if you turn your heads and get a neck cramp for the next 10, 15 minutes. Okay, it's okay. So 11 of those genomes were used for Avian Phylogenomics Project, which I co-led with Goji Zhang and Tom Gilbert. And we used those genomes of 48 bird species to infer a family tree of birds and did a lot of rearrangements to that tree, including some of my favorites, the vocal learners. And we concluded from this data that the birds evolved quite rapidly into many new species after the extinction of dinosaurs, shown by this gray hatched area here. And all orders of birds seem to form roughly around 50 million years ago in a 15 million year period, which was surprising. And there were 2 or 3 independent gains of many different traits we were looking at, including my favorite, vocal learning. So we learned a lot from that, but when students in my lab, including some sitting here, went and started looking at individual genes in those assemblies, there were lots of mistakes. They were good for inferring a phylogeny of a family of species, but they weren't good enough for doing functional studies in the laboratory to manipulate genes. One of my students here, Hannah, spent almost a year recloning a number of genes that we were interested in studying. That caused me, along with the leadership here of the G10K, asking me and others to do what we did with birds with the whole G10K consortium itself, but we had to go one step further. We had to improve the sequencing quality or the sequencing genome assembly quality. And shown here is one of the projects we worked on where we took one of the bird species, a hummingbird, another zebrafinch commonly used in neuroscience, and apply all different kinds of sequencing technologies to it, like PacBio, nanopore, Sanger sequencing, so forth, and scaffolding approaches, which take pieces of DNA and pull them into larger and larger pieces, like bio-nano optical mapping, dovetail high-C approach, and phase genomics and IRMA high-C approaches that look at 3D interactions between chromosomes and have this long-range information, which I'll show you about in a minute. And the take-home message here that we learned over this past 3 years, some published by some others, is that all assemblies we generated here in these long blue bars, giving you contiguous pieces of DNA with no gaps in them, all occurred only when you use long reads. All assemblies using short reads could not get these long, long molecules of continuous sequence with no gaps and hopefully no errors. To scaffold them into chromosomes, the long reads and short reads didn't matter much. What mattered more was the algorithm and the scaffolding approaches I've just mentioned about. And what we found, in showing in this case for the hummingbird, is that with good curation of this assembly, we can get all chromosomes that match the sizes of known chromosomes of the hummingbird in this case. So that was quite remarkable for us. And here are some examples. Here's a gene that we study in the lab. The white signal here shows upregulation of this gene in brain regions for producing learned vocalizations in songbirds, but not upregulated in other brain areas when the animal is moving around, for example. All right? So it's only upregulated in the speaking-like brain areas. And we were wondering, what in the world is happening in this gene in the vocal learning species? And we found that the regions of interest in this gene were not assembled in the prior genome assemblies. These spaces here are gaps. And we found that around those gaps, there was highly repetitive sequence. And looking at the old sequence here, we can find that these different repeat regions labeled in R are really different repeats of different haplotypes, the mother and the father chromosomes that are different. And we could only resolve this by the long reads getting through the entire repeat structure. And what it was is that only vocal learners had this repeat DNA. It wasn't junk DNA. It was DNA that we think was causing these genes to be specialized in speech-like circuits. And so with this new advancement, and we want to do more, but we came to the point where we said, okay, we got to stop. We're satisfied with some improvements here. Let's start a project now. So 3 years later, almost 2.5 years later, that is, we decided we're going to do what's called a reference Vertebrate Genomes Project. Not just 10,000 species, we initially started with the G10K, but all 66,000 vertebrates at high-quality genome assemblies, starting with all orders of vertebrates, roughly 260, all families, 1,000 of them, eventually genera, and eventually all species. And first phase would be our proof of principle project, which we're announcing today. The second phase, I call it the rocking and rolling phase, all right, where if we're starting to do thousands of genomes, then we're really in business. And the third phase, if we complete roughly 10,000 genera, we reach the G10K milestone. And finally, eventually all species associated with other consortiums like the Bird 10K group or the Bat 1K group, also represented here today. So the approach we're using for phase 1, all right, is to have these long reads, PacBio long reads in particular, with 10x genomics, bio-nanogenomics, and ARIMA genomics, scaffolding approaches to pull those, what we call these contigs together into the scaffolds using longer and longer and longer range information. And to give you an analogy, a short read is like the height of a man or a woman, all right? Whereas a long read is like the height of the Empire State Building or even bigger, all right? Whereas the scaffolding approaches are like trying to take these Empire State Buildings together and bridge a continuous set of buildings between Earth and the moon. Right? And what does that gain you? An example is shown here as to what these differences or these longer reads gain you is that if you're trying to assemble a genome that has lots of pieces and all your pieces are small, in the case of this, and I think you're kind of guessing what animal it is, but it looks a little strange. This wing here is in the wrong place. The face is turned around. It's inverted. There are all these gaps in here that can have some errors in them. Long reads makes it easier to assemble. And then with long scaffolding approaches, you can get all the chromosomes together. Now, this would be a perfect genome. We're not there yet, but we're getting much closer. And here is an approach Rong Ray and others in our group have been working on to take ever longer and longer pieces of DNA to string these pieces of DNA into longer and longer pieces until you get chromosomes. And then finally, we'll annotate that is named the individual genes. And we formed a consortium of G10K members and more that here are where the council members are located in blue, and in red are our current genome heads at the Rockefeller University here, the Vertebrate Genome Lab, at the Sanger Institute in the UK, and in Dresden, Germany, represented by folks up here. And what we decided is that we don't just want to do genomes and do them at high quality, but we want to do a focused project that has a scientific mission for each phase of this project. And we took the results that we learned from the birds and from mammals of all orders diverging somewhere in the 50, 70 to million year ago range after the mass extinction of dinosaurs, and applied that across all vertebrates, and in addition to classifications, and came with 260 species that have some common ancestor shared among them that goes back to the time of the last mass extinction, including 58 birds, 52 birds, 58 mammals, some reptiles, fish, and amphibians. And here is a breakdown of the different numbers of species in each category. And what we want to learn from this, once we get all these genomes done, is learn who survived the last mass extinction and why. And that will help us understand who can survive the current mass extinction that's induced by humans. And so with that said, of those 260 species that we selected, today we're announcing the first 15 genomes, representing 14 species. They are 5 mammals, the greater horseshoe bat, the spear-nosed bat, so 2 bat species that are studied for aging and vocal learning, Canadian lynx, a species that was nearly extinct in the northern United States, platypus, which represents traits between mammals and reptiles that lays eggs. We have 3 birds here, the hummingbird that we've improved upon, the zebrafinch as well, the male, the reference, which is now better than the actual reference in the current database. We fixed a bunch of chromosomes with this new assembly approach, but we also decided not to be sexist. We sequenced the female. And one of the reasons why we sequenced the female in birds, unlike mammals, females have both sex chromosomes. And we decided we want both sex chromosomes for all these species. So we're sequencing the heterogametic sex. And a kakapo that's on the brink of extinction as well. Tortoise here, a newly discovered species recently in Mexico. A celian, which is an amphibian that's lost its limbs like snakes have, but independently. And then 5 species, oops, that was a mistake. Don't look at that number 1. And, all right, it's 5 species of fish here, representing different diverse phenotypes. And the cichlids here, like even in Lake Malawi or in South America, are studied as models for species evolution. And my favorites are all here, all the vocal learning animals, so we can ask already right away how these genomes compare to the human genome for speech, even in the first 15 genomes released. And I don't want to stand up here and take the credit for this. Yes, I'm the chair of this organization. I thank the council for voting me in. But it's their work. It's other people's work. And here I list the names of a number of people who are helping lead the efforts for each of these genomes, like Emma Teeling up here, Sonia Vernes in the audience, Tanya Lama also in the audience, Goji Zhang at BGI in China, Bob Murphy for the tortoise. I helped do these bird genomes, and Gene Myers, Richard Durbin, Venki, and others on these fish genomes. And you'll hear from them today, and you can ask questions. What I do want to say is that pick out one species we're dedicating this first release to, Jane the Kakapo. The Kakapo, there are less than 150 animals left on this planet when there were tens of thousands before. They're in New Zealand. There's a rescue effort to help them survive. They're on the brink of extinction. And Jane, unfortunately, just died earlier this year. Today, we're releasing her genome, dedicating this project to her, because her genome is going to be used to try to help save this species from extinction. As much as we can learn from Jane, I think we'll do better with this higher quality reference than what we could with other approaches. So I want to end by giving some more credit here to an assembly working group. This takes a team of people. It takes not just a local team in an individual lab, university, or country. It takes an international team of people studying species from all over the world and using techniques that different labs generate. And I don't have time to go through all the folks here, but some of them are here, and you can recognize some of their faces, the different partners for the different genome groups, and the companies we collaborated with. Even though we're using 4 technologies mainly for the first phase of this project, we work with many different companies around the world. And it was synergistic. They helped us make improvements. We helped them as well. And we're still open to changes for phase 2. You got your council members here. And I'd like the public to know about this costs money. All right? It's not cheap. High quality data is not cheap. So we're looking for people to donate, and people have been donating even before we went live. And here's one donation website at Rockefeller you can get involved in. It's for the entire project, not just for the work that's done here. And we're asking people to donate to particular iconic or endangered species, like the frogmouth bird here, pangolin, mammal, and gharial. Also, only very few individuals left, 235 left on the planet of this alligator. And for the press, you're welcome to the first day of the conference today. It will occur from roughly 1 to 5 p.m. We're going to give updates on specific aspects of this project from members in this audience in this panel here. And I'll go ahead and stop there and open up this session for questions for myself, the panelists, and others we've asked to join us to do some answers. There'll be questions online, questions in the audience that the press have, as well as those who are part of the consortium would like to make clarifications or answer something. Oh, yes, sorry.
So we can take questions.
No. No, make your statements. First statements and then questions. So we're going to have David Haussler first.
Okay.
That's okay.
Thanks, Erich. Let me just say that I'm delighted to witness the launch of the Vertebrate Genome Project. It's a dream. Let me just say that I'm delighted to witness the launch of the Vertebrate Genome Project. My name is David Haussler. I'm the director of the UC Santa Cruz Genomics Institute. And I had the great pleasure of working with these visionaries on the table here. Next to me, Steve O'Brien, Oliver Ryder, and I had the idea that we need to organize the communities that were sequencing various genomes. This was almost 10 years ago. To work together towards a shared technology and an organized exchange of information and mutual support. We created an organization called Genome 10K. The thing that really strikes me is back then, we had no idea how long it would take to get to the point where you had a genome that was complete enough to do science on and could be obtained at a reasonable price. Many of you seen the curves of how the cost of sequencing is going down. But what became very clear, and Erich explained so well, is that what we thought was a genome back then really wasn't a genome that was suitable for doing science. And so it took a long time. 2 assemblathons, I see Benedict Payton in the audience, and the Alinathon that Benedict put together really revealed the state of technology then and revealed the fact that it was not adequate to go forward. So we have been keeping the troops excited about this project to the best we can while furiously trying to improve technology along with many others across the planet. I think we've made a turning point at this point. These new technologies for long-read sequencing and long association over a megabase associated one region with the other are spectacular. And we're thinking of genomes now in terms of haploids. We're talking about reconstructing both chromosomes separately, which is a completely alien concept in the old days, but that is what it takes to get it right at this point. So I am incredibly excited that now we're at a position to get it right, and I really appreciate Erich's leadership. He's been a vigorous and outstanding proponent and visionary for pushing us forward with this project. Now is really the time to get started. I know we were unable to say what a timeline might be in the past, but I think with these technologies, we're hitting the sweet spot, and we've got to get moving. If you look at what the stakes are, we're talking about life on the planet. And in particular, the vertebrate life is so fundamental to everything that we hold dear. It's our responsibility not just to do science, but to also make sure we preserve the life on this planet. And with a technology like this, there's really no excuse for not using it and going out and understanding the beauty of life on this planet. Now it's time to go.
And I'm going to have to stop you at that point. That's great. Thank you. Yes. Next, Steve O'Brien, one of the co-founders of G10K, a former director of the National Cancer Institute, and also Dobzhansky Institute for Bioinformatics in Russia. Steve?
Thank you, Erich. And Dobzhansky worked here at Rockefeller for most of his career. He did, yes. You should go visit his lab, Erich. My name is Steve O'Brien. I'm heartened by the advances of the genome community has dreamed about, stated, and accomplished this day in 2018. When the human genome was sequenced and discovered in 2001, there were about 20,000 coding genes that were pronounced, but at that time we only had names for about 5,000 of them, and we didn't know what most of them did. So when G10K was established in 2008, my colleagues and I had a workshop where we decided we had to write something down. And two of the things I remember we wrote down were that among ourselves we could gather specimens from 17,000 non-traditional species suitable for genome sequences. The 17,000 species was correct. Suitable for genome sequences was not. The other thing that we said was we were going to do it in 5 years.
Did we say that?
Yeah. So those were the days. It's now 10 years later, and the comparative genome sequences of non-traditional vertebrate species, to emphasize, takes advantage, as Erich said, of evolutionary changes, of adaptations, and random genetic drift over millions of years to interpret what this Rosetta Stone means. The benefits we can think of or predict about genome sequences, which you'll hear a lot of today, are dwarfed by the unexpected discoveries we have seen already and must be prepared for in the future. Be prepared for the unexpected, because that's where we are. We are explorers in a new area that will reveal things that we really hadn't anticipated. Since Genome 10K was established as a hopeful consortium working together to unravel genome variety, there's been about 300 to 400 vertebrate species that have said their genomes have been sequenced at some level. As David has mentioned, that level varies from really good, like what's happening today with the VGP, and not so good, which has holes all over it and difficult to do. But that is because they have varying levels of completion, contiguity, assembly, and annotation. The VGP launch today raises the bar for genome utility by facilitating unabridged genome sequences of a continuous, linear, proposed, and looked at sequence of each chromosome of each target species. So not only is the quality of the sequence such that we can actually look at it in a way that somehow resembles the way it is in the species that we ground up to sequence, but secondly, Erich, in his humility, has not stopped at 10,000. He wants to go all the way to 60,000 vertebrate species, which is all of them, not that we can catch, but that exist. So good for you. I think that this is feasible these days. The original promise and dream of the Genome 10K founder, Dr. Haussler, Dr. Ryder, and myself, has morphed from a fanciful scenario to a fulfillment of the motivation to provide a complete and useful whole genome sequence of as many vertebrate species as we can catch. This is our gift to the next generation of genome scientists, whole genome sequences that we can use for all these species. Let's do it now. Thank you.
Great. So next, I'd like to introduce Gene Myers, who's director at the Max Planck Institute for Molecular and Cellular Biology and Genetics. He's also well known for one of the inventors of the BLAST algorithm that we all use and other algorithms that we use in genomics. So, Gene?
Thank you. I'm delighted to be here today. I'm basically at this time an American expat. I'm working in Dresden, Germany, with for the Max Planck Society. I'm also a founder of a Center for Systems Biology there. Our modest little sequencing center, the Dresden Concept Genome Center in Dresden, is one of the 3 hubs, as has been mentioned, for the Orders Project, for the Phase 1 Orders Project. And we expect to be able to fund and sequence hopefully 30 to 40 of the 260 species. So we really, I think we're a small cult. We believe in this thing and we want to move it. So we're putting our money in. In addition, we're also the sequencing hub and also the assembly and annotation team for the BAT1K project, which seeks to sequence the subset of 1,300 species of bats. We've, to the same quality as this project. We've currently sequenced 6 bats to this quality and are analyzing them and hope to have a publication soon. And we will be and are funded to sequence basically a species from every family in the next year, along with selected species to explore the genetic basis of longevity, immunity, and vocal learning. I mean, the bats are fantastic extremophiles, as my colleague Emma Teeling will tell you. We're further funded to sequence a number of European fish to explore the effect of geography on the evolution of genomes, and I'm hoping to sequence several critically endangered turtles towards the conservation efforts, which are also an important goal of this project. All this work is funded, all of my work is funded generously by the Max Planck Society. And the other thing that I need to state strongly is that all of the genomes that we're doing will be sequenced to the standard that we're talking about today. That's a very important thing. And will be part of the open collection of 66,000 genomes that are in the Genome Ark. They will all be released and then be in the public domain as part of this reference repertoire. So I just want to close by, quite a few people have said it, the advances in this long-read sequencing and long-range scaffolding technologies is revolutionizing de novo sequencing. I mean, I basically left the field in 2003, 2004, because with the short reads, I just wasn't, basically producing really high-quality genomes was off the table. I mean, I saw that, and I didn't, as an informatician, didn't want to play. That wasn't my game. But when these new long-reach technologies came in, about a half dozen years really started to come into their own, I saw the potential, and I think several of my colleagues saw the potential for us to finally be able to produce gold standard genomes, things that are as good as the best genomes we have today. I mean, you can basically, out of the box now with these technologies, produce a genome that's as good as the Drosophila genome, which is the best sequenced model organism, release 6. You can get the same numbers of continuity and statistics out of this. So the 15 genomes that we're talking about today, okay, I mean, people have talked about, we're going to do 1,000, we're going to do 10,000 human beings, blah, blah, blah. We're talking about, forgive me, low-quality stuff, okay? What we're talking about are things that will basically, the word was bandied about with the Human Genome Project, things that will stand the test of time. These are things that's going to be one and done. We're going to do these things, and we're not going to have to do them again. Almost everything else that's been done to date is going to have to be done again if you really want to do your science. So I think that, I mean, what's exciting about today is what this is it's only 15, but it's the first 15 of what's going to be a flood. So what's happening today and what we're announcing is the beginning of a trend that's basically going to completely and dramatically alter genomics in the future. Thank you.
Thank you. Thank you. Next is Richard Durbin from the Wellcome Trust and as well as Sanger Institute and Wellcome Trust, Sanger Institute, and Cambridge. He's known for his involvement and leadership in the original Human Genome Project and more recently the 1,000 Human Genomes Project. And Richard, take it away.
Okay. Thanks very much, Erich. It's a great thing to be part of. So my name is Richard Durbin, as Erich said. I'm a professor in the Department of Genetics at the University of Cambridge in the UK and an associate faculty member now at this Wellcome Sanger Institute, which is just outside Cambridge, which is Europe's leading genome institute. So from my perspective, the information in genome sequences plays a fundamental role in biology. And you know, over the last 20 years, genome projects have had a profound impact on the whole of biomedical science, on medical genetics. They've been transformative, and other biotech fields such as plant breeding. And really, you know, we're living in the genomics era, I think, just as people look back at the start of the 20th century as nuclear physics era, people will look back in 100 years' time, this will be the genomics era. And the key thing is it's not over, we're still in the middle of it, and the announcement today is a landmark at the beginning of the next phase of that era. And in that phase, we're going to obtain genome sequence information across all species of life on the planet. And that will change our scientific understanding of and interactions with all biology. And as people have said, some of this has been done, but really at high quality only for a handful of species so far. So genome science has been driven forward by major data generation projects like the VGP, which generate open core information that everybody else can build on, both in academia and in industry and across all human endeavors. And those have all involved international scientific collaborations. And the Sanger Institute has played a key role at each stage. It was founded to play a major role in the Human Genome Project. It's played a key role in each of those. So I think it's that the VGP is a core part of this, establishing these collaborations for the next phase. It's a leading project. The Sanger has now established an internal new program at the top level in sequencing genomes across the tree of life. And as part of that program, it's committed core funds to the VGP. And so far, we've contributed about a third of the data and the computational analysis behind the project. And we really look forward to continuing to participate with all our partners in the VGP. So I wanted to say, I mean, people have said how this is transformative. It's essentially 100 times longer contiguity than the vast majority of genomes currently in the archive. So that's a good number to keep in your head. It's not alone. There are other genomes out there, like Drosophila and human and a few others. But the 15 genomes we're presenting, you know, are in fact a major contribution to that set of things at this level of millions of bases rather than tens of thousands of bases of contiguity. And despite 20 years of work and all the new technologies, there still are great technical challenges in piecing together the billions of letters in a genome in the right order with the goal of no error. And in particular, in scaling that up so that we can do it at the level of hundreds and thousands and tens of thousands that we want to. So it's great to be part of this team. You know, it's great to be back working with Gene, who we worked together 20 years ago, and David and others here, and have new people join. And the team here, in my view, is leading the world in attacking these problems, and it's an exciting time in the field. So,
all right, thank you. So next we have Oliver Ryder, who was also one of the 3 co-founders of G10K. He's at the director at the San Diego Zoo Conservation Institute, and he's our really hardcore conservation biologist here, making us well aware of what species we need to save.
Thank you, Erich. Species for which high-quality genome information is available will have a reduced extinction risk. That, to me, is one of the major recommendations of the Genome 10K Vertebrate Genome Project. As imagined at the start in the then outlandish proposal to produce high-quality whole genome sequences of thousands of vertebrate species, the resultant information would transform all realms of biology. As the scope of the Genome 10K VGP project enlarges, the initiative to sequence species for which such information could be vital to their survival becomes an opportunity of our time and a chance to intervene in the process of extinction. Studies of extinct species using ancient DNA technology have underscored the risks of accumulation of deleterious mutations arising as a result of genetic bottlenecks, inbreeding, and other impacts of small population size, such as genetic drift. Our field aims to move to a more precise and predictive science of extinction risk assessment, utilizing analysis of genome-wide variation. For these studies, a suitable high-quality reference genome is crucial for assessing the load of deleterious mutations and the impact of inbreeding. Previously, it has not been possible to directly identify and understand genetic load in populations. Comparative genomics of primates, elephantids, and bears, to name some of the examples, have already given insights into these factors. And as we announce more genome sequencing, the intention to better estimate extinction risk through genomic studies is building momentum. Studies at the San Diego Zoo Institute for Conservation Research on California condor genomes has advanced to the stage that the California condor is the first U.S. endangered species recovery program to base genetic management recommendations on complete genomes of the founders. A lethal recessive genetic disorder is present in California condors. Called chondrodystrophy, whole genome sequencing information now allows identification of birds that are carriers for this lethal trait. Another critically endangered species, the Hawaiian crow, the alala, has had a high-quality genome assembly produced that, because genes of interest are complete in the assembly, allows studies on behavior and disease that are part of recovery efforts. Some of the most critically endangered animals of our time are being studied with the hope that the knowledge derived from genomic studies can help humankind be better stewards and preserve for future generations a richer heritage of biological diversity.
All right, thank you. All right, next we have Adam Phillippy from the National Human Genome Research Institute. And I'll just say, well, Adam is chairing the assembly group. He's putting a lot of effort and energy into that. And I'd like to say that my let's say, induction into the genome world, I'm a neuroscientist, was really due to Adam when I was trying to play around with a parrot genome one time. And he came up to me after a conference and says, Erich, I really want to help you. And
meant in the best way.
We saw eye to eye, and that's how we ended up partnering up over 10 years ago. So, yes.
Thank you, Erich. Well, welcome, everyone. It's great to be here. Some of us, we see each other every Friday for our assembly group conference calls, and it's no substitute for an in-person meeting like this, so it's great to be here. As Erich said, I'm at the NIH in Bethesda, Maryland. I run the Genome Informatics section, which is in the intermural research program at the National Human Genome Research Institute. And my lab specializes on computational solutions for hard problems in genomics. And one of the hardest problems in our experience is the genome assembly problem, and that's what we've been specializing on for most of my career. And this is a problem that's necessary because we haven't yet figured out a way to sequence entire chromosomes in one pass. Instead, we get fragmented bits of them, The sequencing process serves as a good shredding process to tear your beautiful genome into little tiny pieces. And like Erich showed, the analogy is often having to reconstruct a jigsaw puzzle from those tiny pieces. With the exception, instead of your hummingbird, it's a million-piece puzzle. And there's errors on each of the pieces, and you don't have a cover of the box to look at when you're putting the genome back together again. So it's a bit of a challenge. And when the Genome 10K project was envisioned 10 years ago, to do one of those genomes to the kind of quality that we're doing them at now would have been millions of dollars and years worth of effort. There was entire teams of people at places like the Sanger and the Broad that were dedicated to individual genomes like this. And so it's satisfying now to have the opportunity to take something that was once so much effort and reduce it down to a span of, say, a week per genome. And we're really now at that point with the introduction of new sequencing technologies that we can take what was once an infeasible challenge and accomplish it to great quality. That has a downside that, unlike before, where all of the costs went into the sequencing, now the costs for the analysis really outweigh the costs of the sequencing. And we really risk drowning in our own data. The machines turn off terabases of sequence at a time, and they have a tendency to pile up waiting for somebody to look at them. And so that's a challenge we're really struggling with now. How do we take that wave of data coming off the machines and turn it into these reference quality genomes? So to help attack that grand challenge, we've assembled really a dream team, in my mind, of genome assembly experts here at the table with me, in particular Gene Myers and Richard Durbin, who I grew up reading their works in the field in genome informatics, and it's great to be teaming with them now and hoping to make new history together as we attempt to tackle this Vertebrate Genomes Project. So it's really a special honor to serve as the chair of the VGP Assembly Group and get to think about these problems with them on a weekly basis. That group includes Gene's whole group at Dresden, Richard's group at Cambridge, Harris Lewin's group at UC Davis, Kirsten Howe's team at the Sanger Institute, all of our collaborators all over the globe, and in particular, Henri and my lab at the NIH, who's been really instrumental in dragging these 15 genomes across the finish line in the past couple of weeks. I'd also like to recognize our corporate informatics partners, particularly at DNA Nexus and Amazon Web Services, who have done a lot to help get this project off the ground. Amazon Web Services has generously provided free hosting of all of the VGP data on a cloud S3 bucket that we're calling the Genome Arc, and all of the raw data as it comes off of the machines, as soon as it can, is uploaded to that central resource, which has enabled us to collaborate and share this data with our partners. And DNA Nexus also have helped us scale our processes, putting a lot of these assembly processes into their cloud platform so we can really scale from one genome at a time to doing 10 genomes at a time. I remember back in April when we started doing the contagging for this first set, at one point we had kind of 10 running in parallel, and they all finished within a week, so that's really helpful. So to conclude, we've really built a fantastic team here. I have high hopes and expectations that we'll be able to achieve these goals that we've set out. And this is especially important for those species, as Ali mentioned, that are soon to be gone from the Earth. So I look forward to providing a full update on our group's activities in the plenary this afternoon, and thank you all for being here.
All right, next up we have Emma Teeling, who is our BAT1K expert with Sonia Bernes here, and at the University College in Dublin. And I can assure you she's not the only woman in leadership, all right? But she is a good representative of the whole team here. So go ahead.
So it's great to be here. I'm a professor of zoology at University College Dublin in Ireland. And I just want to set the scene a little bit to think about why today is so extraordinary. So this year marks the 75 years since Ernst Schrödinger wrote his book, What is Life? And in that little book, he mused about what was the raw material of inheritance, what potentially was DNA. And he's a physicist who moved into biology to think, well, it had to be some form of aperiodic crystal, and there had to be a way to try and maintain this crystal, but yet they didn't know how it was replicated. And this little book spawned a lot of the ideas of Watson and Crick and Rosalind Franklin and Mark Wilkins, and the scientific consciousness moved towards discovering what DNA actually was. But that was only 65 years ago. And there are people alive today here in this audience who were born earlier than that, I am sure. Not me, of course. But think about it. 65 years ago, they didn't know really, was it the nucleotides, was a protein? What was the raw material of inheritance? And now, today, we're able to sequence the entire genome from multiple different species, like this weird Sicilian. I know Mark Wilkinson won't agree with me, but we're now able to go and look at all of life. We're able to uncover the raw material of inheritance. We're able to then use that information to make trees of life. And now with these new exquisite genomes, 15 we're talking about today, we're able to uncover maybe what are the genetic bases of the rare, weird adaptations that these extraordinary animals actually have. And so this is a landmark event, because in 65 years we've come along so much. So in 2009, when Steve asked me to come to Santa Cruz to talk about the idea of Genome 10K, which really was quite outrageous, because we didn't have the methodology yet, we didn't have the DNA, we didn't know how we were going to sequence it, but Lewin had come on the scene, and now it was possible maybe to sequence these genomes. Within a decade, we realized that we were going to have to get much better types of DNA. We had to try and work out how could we assemble and move things forward. And as a group, we brought together mathematicians, geneticists, conservation biologists. We brought together zoologists, people who actually know these animals, where they exist, what's their unique lifestyle, and also with access to the DNA. And really, G10K spawned VGP, spawned BAT1K, spawned the Earth Biogenome Project that Harris is going to talk about. And that today we're now able to uncover really the blueprint of all different species. We're able to look at each different base pair, potentially assign it to some type of functional aspect. It's unique. And for me, as part of BAT1K, the idea that we could actually now go and sequence the genome to that level of exquisite definition of every living bat on this planet, which I think everybody should be working on personally. They're the most extraordinary mammals. We're going to be able to uncover what is the genetic basis for flight, for these long, healthy lifespans, for some type of immunity against different viruses such as Ebola. They don't die from Ebola. They don't get cancer, and it's all in their genomes. And now we're going to be able to uncover it. But it took a decade from 2009 till now to get to where we are. But I think that you have to, for good things come for people that wait. And we move the field forward, and this is just the start. And the floodgates will be opened. And why should we not now sequence all of life to see how it's evolved on this planet? And so I'm very glad and honored to be here, and we can talk about bats anytime.
I have to say I'm getting inspired myself listening to you folks. Last but not least, have Harris Lewin from UC Davis, well known for studying chromosome evolution, how chromosomes rearrange themselves in different species to make trait differences. He's also, like you just heard, leading even a larger group, the Earth Biogenome Project, for sequencing all eukaryotic species. Harris?
Thanks very much, Eric. So I'm Harris Lewin. I'm a professor of evolution and ecology at UC Davis and a member of the UC Davis Genome Center. I'm also a founding member of the G10K consortium and a lead of the Mammals group. And I also serve as the chair of the working group of the Earth Biogenome Project. And so I've earned the last spot to make comments at this little conference. So for 15 years, I've really dreamed of the possibility of producing chromosome scale assemblies en masse for our studies of chromosome evolution and the role of chromosome rearrangements in adaptation and speciation. 15 years ago, when we began to think about this, and when Steve invited me to the first meeting for G10K, I was the only one there, Steve, who was calling for chromosome scale assemblies. And I was supported by Steve. We even got a few sentences in the original paper. And the tremendous advances in technology now, long read technology and scaffolding techniques and informatics, we're there. And Erich has really brought all this together in a way that has never been done before. So today, after 15 years, the time has finally come, but of course it's only the beginning. It's the beginning of producing thousands of reference quality genomes, not only for the G10K group and the Vertebrate Genomes Project, but also for many other taxa, including plants, other animals, fungi, and the microbial eukaryotes. This first effort to produce reference quality genomes at scale is a critical step for the Earth Biogenome Project, which, as many of you know, aims to sequence the genomes of 1.5 million eukaryotic species in 10 years and approximately 10,000 reference quality genomes for all eukaryotic families in 3 years. It's going to cost about $4.7 billion to do the Earth Biogenome Project, but that in today's dollars is less than the cost of sequencing the human genome at its time. The G10K consortium has been leading the way among all of the scientific communities that I interact with, the Earth Biogenome Project, and what we're learning is essential to reaching our ultimate goal of sequencing all of eukaryotic life on Earth. It's outlandish, it's outrageous. It was as outrageous as what Steve and Ollie and David proposed 10 years ago. But it's feasible, and it's inevitable. While much remains to be done, these first 15 vertebrate genomes are a statement to the world that what we aim to accomplish is in fact feasible. The VGP has shown that it's possible to obtain samples from all over the world, isolate high-quality, high molecular-weight DNA and sequence that DNA, produce physical maps, integrate all the data from the 4 methods into an automated assembly pipeline, thanks to Adam and Arang, curate those assemblies, and prepare them for submission to public databases so that they are freely available to the scientific community and to the world. The impact of this first release of sequences, 15 sequences of vertebrate genome assemblies, will immediately impact the field of evolution, comparative genomics, organismal, and conservation biology. The scientific impact will be greatly amplified by completion of the Ordinal project that Erich is aiming at, about 250 genomes in all in the coming year, and I am now, for the first time, confident that this can be achieved. I want to congratulate Erich and the entire production and assembly team on this magnificent accomplishment.
Thank you, and I'd like to thank all panelists. So we're running 12 minutes overtime, but there's still time for question. I'm going to go ahead and extend it even 5 minutes beyond the hour for those online and others who want to ask questions. And you can ask questions to us here, the technology partners in the room, and the others who couldn't fit up here. And I'd like you to stand up, who are part of leadership. Rong Ray, who you heard about at NIH. Paul Flisick, who's director of the Ensemble Annotation Team. Federici Di Palma, who's director at the Earlham Institute in the UK for Genomics. And they're here to answer questions as well. And our technology partners stand up as well. Okay. All right, go ahead. So Sid Severez from Arima Genomics, Jonas Korlak from PacBio, and I'm blanking out. Zev, yes. Go ahead. Yeah. So Zev Korno from Phase Genomics. All right, so questions? And somebody will, Sadie, or who's going to be taking the questions from online? Okay, all right. And just to say, we have about 6 folks here from the media in the audience, and double to triple that number online. Did we say everything that was needed to say? Go ahead, yes.
Hi. So as technology, it's very loud, sorry. So as this technology is advancing, and I think it's
Could you
wonderful.
Introduce yourself as well?
Sure, I'm Jeannie Garbarino. I work here at Rockefeller. So my question really is around the idea of sort of the responsibilities that we as scientists need to recognize when we're developing such technology that can be so impactful. And so one of the major ways that I'm wondering if there's a connection or impact is on the field of synthetic biology. I know that there's a lot of efforts out there to write chromosomes in vitro, and having these sequences will certainly allow that technology to be expedited in some way. And so are you considering the implications on an ethical basis in terms of having this technology available and how it might impact other types of things related to DNA technology moving forward?
Maybe I can ask Jonas to answer. Yes, since you're a technology company. To take the microphone, yes.
My name is Jonas Korlach, and I'm Chief Scientific Officer at PacBio, and we're the underlying technology that was used for the assemblies that are then used into the scaffolding. And not speaking for the company, is my personal opinion that there is definitely an ethical component that's already also been discussed one way or another. I think there's still a gap, but nevertheless, we need to be conscious that almost with any scientific breakthrough of the magnitude that is presented here come responsibilities that we need to consider. And so I think there's tremendous opportunities to preserve species from extinction and perhaps even bring back some species from already extinct species and take that information when the habitat has been restored to try to bring those species back. I'm a believer in driving technology with an appropriate awareness of the risks and the ethical responsibilities and not shy away from them and from that progress because of those risks.
Anybody like to add? Go ahead, David. Okay.
Yeah, so I think this is an important area. We know from, at each of the stages in genome projects, there's been a discussion around the ethical side, and from the original Human Genome Project, the LC, ethical, legal, and social implications alongside. I think in the broader context of the EBP, there's actually two sides to this. One is the use of the data, and the other is the benefit sharing across the various parts of the world where a lot of these organisms are and are coming from. And there's a very active component to that.
EBP standing for Earth Biogenome
Yes,
Project.
the Earth, which Harris talked about. So I think it's the issues you raise are part of a broader discussion around the use of genomic information. As a scientist, my belief is that the right thing to do is to generate the information and manage the framework in which it is used through a combination of legal and other societal and scientific approaches. And historically, many of our institutions have been very involved in those things. So that's the context from my perspective.
I think Gene was. Okay, Ali and then Steve, and then we'll get the next question. Good.
Well, I agree that this is a societal question, but I would point out that this started a long time ago with the gene splicing, and there was an Asilomar conference where the potential impact of moving genes between species was discussed, and I believe there's an inevitability in our quest to understand life that's going to elaborate the genomic sequence information of species, and this will give the current and future generations of humankind an unprecedented ability to design nature. I think that it has to be part of the societal discussion and education that how do we, that is done with intentionality, not by acquiescence or not paying attention. So I think it's an important issue. It affords great opportunities for humankind, and it also places us in a role that's ever more clear, that is unique in the history of life on the planet.
All right, Steve?
Yeah, I kind of agree with Ollie, but I'm reminded that there's a difference. I agree with the questioner that there certainly is an important issue about the ethical concerns about what we should and should not do with the information that we develop and make with all these species and with all these genomes and with the technology about gene editing that's just stipulated at the beginning. But I do know that there has been in our society and in our culture and in our communities a emphasis on protecting humans from this, and that's why we have an FDA that allows drugs. But there's another precedent, which is if you look at veterinary drugs, there's almost no sanction at all against putting out a useless drug in the veterinary community. There's no regulation to speak of whatsoever. So we could start worrying. There's a big discussion in human genetics and human medicine about using CRISPR and gene editing techniques. But what about doing it in some little bat that Emma studies that nobody cares about? Is that illegal? Is that ethical? Should we be discussing it? I think the G10K community and this conference and the future should be thinking about the points Ollie made. These things do have consequences, and we do need to have some, at least discussion, if not some standards commuted, or we will make the same mistake that the dogs and cats have suffered because of all of the worthless vaccines and drugs that are out there. Okay.
All right, great. Is there any question from online?
We do have an online.
Okay, go ahead.
I think this is on from Liz Panisi from Science. Liz wants to know, please clarify what the data usage policy will be for these 15 genomes. Are there restrictions? What will be the data availability going forward?
Okay, I guess I'll take that question, and then if anybody else wants to add to that. So yes, we had a lot of discussion, a lot of vetting of a data use policy for these genomes. And you can imagine what all the effort the folks up here at the table, some in the audience here and others not here, have put into generating this data and then making it publicly available and then somebody else scooping them. So we had to avoid doing that. But at the same time, lots of my colleagues are saying, Erich, please share this new zebrafinch genome that you have because the one we're using we're not happy with. And so the data use policy basically is anybody can use these genomes in the scientific community or otherwise to publish papers on one gene across species or 5 genes within a species. And they have to notify me as the chair. And any kind of studies they like to do comparative genomics across species before we publish a major issue of papers on the 260 species, which we expect to be in roughly 2 years, that's what we hope, they have to contact me or become part of the consortium and collaborate with us. And we think this is a fair policy. We vet it with different journals. We vet it with the annotation centers, the public ones, and so forth. And we'll see how it works. So far, something like this worked decently for the avian phylogenomics project. I think it's going to work for us. David?
Just want to clarify that the data will be available, though.
The data is available.
Openly, immediately.
The data is available today. Okay? Well, 8 a.m. tomorrow, actually. Yes, yes.
Tomorrow.
Yes. Openly, with that embargo policy for use of it. Anybody else want to add to that? Okay, question here? Good.
This is related to that. I didn't hear, I'm sorry, I didn't hear how long it was before you published.
So what we did as council, so this is a policy we voted on, we vetted with other groups as well, Sanger and so forth. So we came to some kind of balance of timing. All right, and what we did from the day of the release of the genome, 2 years from that day, so 2 years from tomorrow.
And then you can only work on 5 genes within a species for 2 years?
Well,
or
you can
publish?
you can publish.
Right,
with some
okay.
kind, of course, with credit still being given to the generators of the data,
Oh,
right?
yeah, yeah.
But if you want to do more than that, you can work on it, but you got to wait to publish
years.
that. 2 years.
Okay.
Yeah, until we do.
Just
I'm
I
hoping we're going to do it sooner. Yeah.
2 years or until they are published.
Yeah, 2 years until they're published.
Or,
No,
or
I know
until,
that.
whichever is first.
Yes, whichever is first.
You have 2 years to publish, otherwise
That's right.
it's free game. Okay, thanks.
Any other?
One comment on that.
Go ahead, go ahead.
As I said when I was giving my spiel,
Talk a
that
little, okay.
the data goes up and is visible to anyone who cares to download it, even the raw data, as soon as it comes off of these sequencers. And to just emphasize that we're talking about 15 genomes in this release, and by our last count of vertebrate genomes, there's maybe only about 20 in the current public archives that meet the kind of standards that we're aiming for. And so we're almost, in this first release, 15 might not sound like a big number, but for the past 2 decades, in 6 months worth of work, we're roughly doubling the amount of high-quality genomes that are available. And I just wanted to make that
And I would say 2/3 of those 20 genomes were generated by members sitting up here at this table or part of the G10K project. Yeah. All right, we can take one more question. Yeah, it's coming now, yeah. Was there any other questions?
So, thank you. I'm Giulio Formenti from the University of Milan. I was curious about your species concept, the definition of species, if you, because of course that's an important question if you want to have all of them sequenced. And also I'm aware, you know, that there are a lot of cryptic species, so I assume that this effort having reference genomes will highlight that there are so many more species maybe out there that we don't know about if you just look at the phenotypes, let's say. So
Yeah, I'll take that as well, because you know, I've written about this, and so one of the things, the number of species keeps expanding and contracting depending on how people classify that species and whether they hybridize or not. And so where do you draw the line as to when you have completed 66,000 species? And I don't think we're going to really know that number, the actual number, as well as the defining exactly what is a species of an individual relative to another until we have enough genomes to actually use the genomic information to decide, is this a different species or not? Yes. Harris?
Yeah.
Then Ali and Steve. Okay.
Yeah, one of the really important aspects of VGP is that they're making chromosome scale assemblies. Many of the species that we're asking questions about, is it a species or a subspecies, they don't even have a karyotype. And so these chromosome scale assemblies are really important in terms of knowing the genomic organization, which is, in many cases, in addition to other types of measures, critically to determine whether it's a different species or not. And so I think this is going to go a long way to help resolving some of those questions around speciation.
Ali?
Already, the emerging field of genomics is causing a revolutionary change in our understanding about the ancestry of species and their sustainability. And I'm referring to post-reproductive isolation gene flow hybridization events. So 10 years ago, we didn't know that that 2 sister species in the genus Homo were interbreeding with the ancestor of modern humans. That was an initial example. There are many now examples that are coming out with great frequency that really are humbling in the way that they show how we didn't appreciate very basic aspects of the way evolution of life occurred on Earth. And the definition of species, similarly, is going to take a whole new conceptual analysis as we have these whole genomes and studies of variation within species.
Richard, then Steve, go ahead.
Yeah, I just
Steve,
want
you'll
to
get the
echo
last
that. I think there's an increasing realization that there's gene flow and exchange between what are currently defined as species, and our understanding of the concept is going to change over the next 5 years or so and is in the middle of changing. And to some extent, I think there's an analogy with about 20 years ago, there was a lot of arguments about what a gene was and how many genes there were, and it was the big question. And now, actually, that's not really a major question in molecular genetics anymore. You might, and I think once we have laid before us the genetic structure of life, we will still talk about species, but we'll be more comfortable about it. Unless, yeah. But
Steve,
it's certainly...
Steve gets the last word for the Q&A. Go ahead.
Oh, I disagree with all these guys. I actually think that Linnaeus got it right most of the time when he named all the species of vertebrates, and that we have been fine-tuning that for a while. There is no right or wrong definition of species, and there will always be perturbations of what goes on and what changes. But I also, like Erich and Ollie, have written about this as well. And I'm coming around to the conclusions that the species concepts that we've had all along are pretty good, and that the fine-tuning of them, even with genomic data about hybridization, which is real for sure, but it's only in incipient species that are about to happen, isn't going to change much. So I wouldn't worry about it.
All right, I'd like to thank all the panelists up here and down.